Hi, my name is Ali Memon, and I go to Interlake High School. I'm in 11th grade, and this is my first year at Bio Expo as well as my first real science fair. And I knew I wanted to do lab and research because that is what interests me the most. So the hard part was when I wanted to decide what to research specifically. So I was reading an article during the time of the Ebola hype, and the article mentioned the possible use of silver nanoparticles as a means to treat or even cure the Ebola virus. So this got me interested into silver nanoparticles, and then I did some more research about silver nanoparticles, and I found out that they were much more simpler than I thought they would be, and I was really intrigued by silver nanoparticles and how widespread their uses are. For example, Samsung uses them. They're used in catheters, and many like silver spoons and silver doorknobs self-sanitize due to the oligodynamic effect, which is a more formal name for how silver nanoparticles are antimicrobial. So then I researched how specifically silver nanoparticles kill the bacteria. So I found out that they release these nanoparticles, and then the most prominent factor I found was that they degrade the cell wall, and then that causes the cell to die. Also, the cell can do apoptosis, as well as the DNA has been found. The DNA has been found to be denatured, and also it interferes with the catabolic processes. And then I thought these were the only methods, but then I read an article that used silver nanoparticles on lymphocyte cells, and it stressed the importance of oxidative stress. So what oxidative stress is when there's excess oxygen, and through the many chain processes in the cell, the reactive oxygen species, ROS, forms, such as H2O2, hydrogen peroxide, and many other. And then these were particularly a problem when nanosilver was in the bacteria, because nanosilver had increased the production of the ROS. So I thought this was really interesting, and I was surprised that I had not read about this before regarding bacteria cells. So I wanted to study how ROS, how important oxidative stress was in bacteria cells, since I had never really read anything about that. So my question that I derived upon was, how do different levels of oxygen affect the use of silver nanoparticles as an antimicrobial agent? And what it means to be antimicrobial is the silver nanoparticles inhibits the bacteria from further growing. So what I did was I had one petri dish which was for low oxygen conditions, one petri dish for standard oxygen conditions, and one for higher oxygen conditions. So now I faced a challenge on how to increase and decrease the oxygen levels. So I decided, after talking to my AP chemistry teacher, that I would use hydrogen peroxide in a flask and manganese, the reaction between those inside of a Ziploc bag, to increase oxygen inside of this particular system, and so the bacteria would grow. And then in this one I just left it as is in the Ziploc bag